Okay, we're on the home stretch here. We're on the chapter 16 on DNA. All right, so just a little background. The DNA structure wasn't really determined until the early 50s when Watson and Crick um, they came up with their double helical model of DNA. What we know now is that our heritable information is encoded in DNA and that it's responsible for a lot of our traits, whether it's chemical, physical, or even behavioral. Okay, this is one of their original models of DNA. It was made with ring stands and Tinker Toys. So very, like, I would say innovative for the 50s for sure. Watson's on the left and Crick is on the right. Um, Anyway, early in the 20th century, not only did we not know what DNA looked like, we probably didn't really care to a certain degree because we didn't really know what DNA was used for. And it wasn't until uh, the late 20s, the late 20s when a physician, Frederick Griffith's lab, he actually was trying to come up with a vaccine against pneumonia. So he was working with two strains. One was pathogenic, pathos meaning disease, genic meaning causing. The other was not pathogenic. Same bacteria species, but one had a capsule, i.e. the pathogenic one. One didn't have a capsule, the harmless one. The pathogenic one we call the S strain because it has a capsule on the outside. And the but not so nasty one was the R strain because it didn't have a capsule. So take a look here. What they did is they took living S, or what you can do is take those living S cells, inject them in a mouse, and the mouse dies. Okay? Dies of pneumonia. If you take the R strain, inject it into a mouse is fine. Okay? But that's a problem because, utilizing the R cells probably isn't going to give us a very good vaccine against pneumonia. So what we can do is we can take the S strain and we can kill them with heat. We can boil them, inject them in the mouse lives. Okay, great. But we don't really know how good of a vaccine that would be, especially when we have dead things in us. Maybe we could add a living thing that's kind of like active and kind of get the attention of our immune system. And maybe, that would be a better one. So we add our heat killed S strain and our R strain, mix them all together, and swing and a miss. The mouse dies. And wait a minute, we do our little mouse autopsy, we find out that living S cells are in there. Even though we never put any living S cells in there, somehow they were. Well, Griffiths said, you know what, I bet there's some sort of transforming principle that is in those dead cells that kind of jumped over into the living cells, changed the living cells into the S type, and then went from there. So it wasn't until later, another lab, the Avery lab, in the 40s, what they did is that they actually figured out that the transforming principle was DNA. They took two enzymes, a protease, which dissolves proteins, and a nuclease, which dissolves DNA and RNA. Or, I'm sorry, not dissolves. It actually chemically breaks them down. They're two enzymes. So you do the same final experiment where you add a protease, where the protease breaks down the proteins, and you still get the death of the mouse. But if you take the nuclease, that breaks down the DNA, and then at that point there's no transforming principle to go from the dead cells to the living ones because so they concluded that transforming principle was DNA and hence our molecule of heredity. Sadly, it really wasn't. People didn't really look at that experiment and go, yeah, that's right, DNA, it does it. No, and it actually wasn't until later when there was a lab, Hershey Chase, in the 50s. They did experiments with bacteriophage. So that's just a virus that kills bacteria. So here's a picture of one, and then here's a bacterial cell. You can see they're kind of on the outside of the bacteria, and they're trying to kill it. Anyway, the 50s, they did an experiment we call a blender experiment. So what they did is they took radioactive sulfur and radioactive phosphorus, and they grew bacteriophages either in sulfur or phosphorus. Okay, so protein is made out of nachos, right? Nitrogen, carbon, hydrogen, and sulfur. Okay, so the sulfur, the radioactive sulfur will tag the proteins. DNA, on the other hand, is made out of poncho, phosphorus, nitrogen, carbon, hydrogen, oxygen. All right, so the radioactive phosphorus will tag the DNA. All right, so we grow this, and we got an Erlenmeyer flask, right? And we put it in a blender. That knocks anything off the cell, just kind of knocks it off. And then we do a centrifuge, and the centrifuge spins down the cells into a nice little pocket, and at the bottom of this test tube. And then at the top, we have liquid that's on top. We call it supernatant fluid or just supernatant. And then that little pocket that I mentioned is the pellet. So what they found is that, when you do this, the supernatant fluid that's in the radioactive sulfur one, that ends up being radioactive. If you take a Geiger counter, you check it out, that's radioactive. Well, you do that the same with the radioactive phosphorus, and it's the pellet down here. That's the radioactive part. So what that's telling us is that proteins staying on the outside of the bacterial cell, DNA is going on the inside. And the idea is that when that DNA gets inside there, it gives directions to the bacteria cell to make more virus parts. So protein and DNA, and then they assemble it, make the virus parts, and then they break out. So ultimately, we kind of read this as DNA is a molecule of heredity. Some other interesting experiments or just data. We have Chargaff's rule, where he noticed that that any it doesn't matter what, but you always have an equal number of adenines and thymines in DNA, and you have equal number of guanines and cytosines in DNA. No matter what species, no matter what organism, they're always the same. So if I have 10% adenine, I'm going to have 10% thymine. And that tells me I'm going to have 40% guanine and 40% cytosine. All right? Because that would be 20% adenine and thymine and 80% guanine and cytosine. Okay. Well, and here you can see kind of the structure of DNA, sugar phosphate backbone. So phosphate, sugar, and so on. We have our nitrogenous bases from DNA, A, T, C, and G. Anyway, so Watson and Crick were, they were using some of their data. They were using kind of Chargaff's rules, and they were kind of working on their model of DNA. And they were talking to Maurice Wilkins, and they were like, we have this problem, and Wilkins. Depends on whose side of the story you're listening to. It's really hard for me to kind of understand 100%. But basically, Wilkins went through Rosalind Franklin's desk and grabbed her data and said, "Oh, I've got this photo right here, that Rosie did of DNA crystals using X-rays, and this is Rosalind Franklin on the left, and her picture on the right, Rosie, and then I think this is called Photo 51. So the idea is that you're looking kind of, I think it's lengthwise on the DNA. But anyway, ultimately, Watson and Crick looked at this, and so it's told, they looked at this, and that was kind of the missing piece they needed. And what's ultimately sad is that Rosalind Franklin, because this is 1950s, early 1950s, she really didn't get any credit for this. And as a matter of fact, if you read Watson's kind of story of all this, they just make it sound like she's mean and nasty and not easy to work with. And who knows, maybe she was. I kind of doubt that she's as bad as everybody kind of makes her out to be. But ultimately nobody really asked her, hey, can we borrow your data? Can we use your data? Can we put you on as an author in this manuscript that we're putting together? Hey, can we share our Nobel Prize money with you? Although I think that she had actually passed away prior to them getting the prize, and this is an example where somebody did a lot of the legwork, responsible for one of the biggest breakthroughs in the 20th century, and got no. The only credit she really got early on was just probably a lot of hate and discontent. So, definitely a sad story. Anyway, so here's just a refresher of kind of DNA structure. We have a sugar phosphate backbone, and then in the middle we have our nitrogenous bases that hydrogen bond together. So each strand is covalently bonded kind of vertically, but then they're hydrogen bond in the middle. So what that means is that we can pull the middle part. We can pull the middle apart and put it back together again. Anyway. And I think that's, since we've already gone over DNA, this is really all I'm going to say about like its structure. This is showing kind of like part of how they were using that data, where they kind of figured out like we put our purines together, two purines, i.e. An adenine and a guanine, we put them together, they're not going to fit the width. If we put our two pyrimides together, i.e. cytosine and thymine, it's going to be too narrow. But if we put one and one together, boom, we actually get, it's actually works out. And then they use Chargas rule to where they realize, hey, like adenine is always equal thymine, cytosine is always equal guanine. Really kind of an amazing feat how they did take all these like forms of data. Especially, it wasn't like there was like the internet flying around. It wasn't necessarily easy to get all this information collected, and then but they did. So, and that's kind of a really, really super condensed story of how the structure of DNA was discovered.